Hello, and welcome to Glycan Hub, the podcast in which we explore health, disease, and longevity through the lens of glycobiology. My name is Irina, and I'm your host. In today's episode, we'll tackle the question on how much of our health is the product of our genes versus our environment. And to answer this question, we'll be investigating the glycans on one very special protein produced by the immune system and known as immunoglobulin G, or IgG for short. Now, IgG is the most abundant antibody in our blood, and the glycans present on this antibody have been extensively studied in context of disease and aging. My guest today is a geneticist who has authored numerous papers on immunoglobulin G glycosylation. Her research interests concern genetics of complex traits, including glycosylation and involvement of glycans in the regulation of immunity. We'll discuss genetic mutations, pleiotropic diseases, and whether there is such a thing as bad glycans, as well as some of the open questions in the field. For instance, whether immunoglobulin G glycans are causative of disease or change as a consequence. My guest today is currently studying regulation of antigen-specific IgG glycans and connection between immunoglobulin G as well as other plasma protein glycans in human diseases. She conducts her research at Genos, the leading laboratory for high-throughput glycomics. A warm welcome to Olga Zaytseva. Hello, Olga, and welcome to our podcast.
Hi, it's really nice to be here.
And we are very happy to have you here. You have a background in genetics and molecular biology. Why did you choose to focus your recent research in glycobiology?
Basically, because there are a lot of unanswered questions yet in the field of glycobiology, specifically when we are speaking about genetics. Not too many people are doing that, which leaves you a lot of ground to cover. So it's really exciting. You're able to do something completely new.
Absolutely, go into a field that's maybe less popular than the mainstream.
Yeah, indeed.
So we actually haven't, I realized, dedicated a whole episode to genetics of glycosylation or indeed epigenetics. We are going to be focusing mainly on genetics today, but could you, for a beginning, maybe define the two terms? What are genetics and epigenetics?
Well, those both are very broad terms, to be honest. So genetics is like the branch of biology which studies inheritance of traits. So why we look like our parents or why we don't look like them. Because we also know that sometimes, two people with brown eyes can have a kid with blue eyes, and then genetics explains that as well. So why sometimes you do not resemble your parents in your traits. And so genetics came into being when in 19th century when Gregor Mendel started his experiments on peas, and then he realized that there is some sort of a unit of inheritance in a way. Now we call these units of inheritance genes, hence genetics. And at that point, of course, Mendel himself didn't know whether there is like any physical material embodiment of the gene. But now we know that genes are encoded in our DNA, which is situated in the nucleus of every cell, almost every cell in our organism. And indeed, it acts as this storage device for the information how to build proteins, which are structural components of our bodies, and they also participate, they do all different functions in our organisms, all enzymes are proteins. Genetics studies all different ways in which these traits, like our phenotype, meaning how we look like, all kinds of traits that we can imagine, how we can pass them through the generations. And epigenetics, so from Greek, epi means above, over, outside. So usually, when you hear something like that, epi something, it's something on top of. So something on top of genetics. Something which is not kind of explicable by traditional ideas in genetics. Like if we want to have a new trait, something new that we can also pass in the generations, we need to introduce a change into the DNA molecule, change the sequence of the letters which encode the proteins. But sometimes this also can be achieved by not really introducing any mutations, but by changing like the way this genome is being regulated, like which genes are active, which genes are not active, and there is a specific code on top of the DNA code. As I already said, DNA is condensed into chromosomes, so it's like packed in a sort of way in the nucleus. And of course, for the genes to be active, they have to be accessible for all the cellular machinery, which does. So we know how DNA instructs the cell which proteins to produce. First, there has to be a transcription of the RNA from the gene, and then this RNA acts as a messenger, so it goes to the ribosome, and the ribosome starts to build the proteins according to what is written in the RNA. So the DNA actually has to be accessible so that the RNA can be transcribed. And there are certain kind of tags or marks in the chromatin and the proteins which are connected to the DNA, like which are the structural basis of the chromosomes. So there are like a lot of different, there is a whole code which kind of determines the state of the chromatin, so to say, and there can also be some kind of marks on the DNA itself, methylation marks, which probably, a lot of people who are interested in recent development in molecular biology are familiar with the term methylation. And it all governs like which parts of our genomes are active and which are not. Because if we think about that, almost every cell in our organism has the same DNA, has exactly the same sequences of like any gene, and but if we look at the nervous cells and the cells of liver, for instance, they're very different. They don't look alike at all. The shape is different, the function is different, the response to any kind of intervention is different. But how do we achieve that? This is because different parts of the genome are active in these different cells. And usually when we're speaking about transformation of the cells into metastatic cells, into cancer cells, it usually also has something to do not only with the physical mutations in the DNA, but in the epigenetic code of the cell as well. It also changes a lot. Methylation of the DNA changes and histone marks change. So the cells start to activate the genes that do not have to be active at this point, and they start to silence the genes which actually would be able to control the proliferation of the cells. So they switch to a completely different new program.
That's an incredibly thorough response. Thank you. But I think just this idea that genes can be turned off and on is very fascinating. And how does this happen in disease?
Well, it depends on which disease we're talking about, and there are like a lot of different ways in which that can happen. So if you're speaking like about epigenetic mechanisms, I think like even depression has a lot to do with epigenetics, because we know that depression is also like a complex disease, and there are a lot of factors which influence whether this person will have depression or not. But there is definitely an epigenetic component to that. A genetic component for sure. There is genetic predisposition to have depression, and there is epigenetic reasons for people to have depression. I think it was shown. So there are two. I mean, I'm not really like an expert in the field, but I do remember that we have like glucocorticoid receptors, and we have like also the levels of serotonin in the nervous synapses. And both of these things can be also regulated on the epigenetic level. So it was even shown that kids who at an early age were subjected to some very stressful situations, maybe their parents were abusive, maybe they didn't have any parents and they have to grow up in complete poverty, like, really small kids, like even before the age of 2. The methylation of their glucocorticoid receptors, if I'm not mistaken, is being changed. And so later on in their lives, when they're already like grown-ups, they feel the consequences of that because they're more prone to depressive episodes, for instance, like that.
Absolutely. We are going to keep touching upon epigenetics throughout this conversation, but I think a good place to start is I'm going to quote something you wrote in one of your studies, which is glycan biosynthesis is not template-driven like nucleic acid and protein synthesis. What do you mean by this, and why is it relevant?
That's an interesting point, because we know that our bodies are built of different types of biological polymers, meaning it's polymer, so it's a large chain, some sort of a chain-like molecule, which consists of sort of building blocks which are repeated throughout the structure. And all main, I would say, classes of biological molecules are built as polymers. DNA is a polymer, stands for desoxyribonucleic acid. RNA is a polymer, ribonucleic acid, which is like similar to DNA but a bit different. Proteins are also polymers, so they consist of repeated amino acid residues. And the thing is, when we're talking about this fundamental thing in molecular biology, central dogma, so-called central dogma of molecular biology. It's about proteins being like the instructions for construction of the protein being inscribed in the DNA. So we have DNA, first of all, which is storing all the information about proteins and RNAs. And to pass this information on, as I said, an RNA molecule needs to be, so the process is called transcription, because it's literally as if you took like a page from a book and you started rewriting it on a piece of paper. So the book is still kept in the library, but you write down everything that you need to know from that. This node will represent RNA molecule, which then will go to the ribosome, and then the process of translation will start. So the sequence of the nucleotide bases in the RNA molecule will determine, like 100% determine the sequence of the amino acid residues in the protein. So all two processes, transcriptions and translation, they are both completely dependent on some sort of a template. DNA acts as a template for the RNA synthesis, and RNA acts as a template for the protein molecule. Although here translation, so we need to decode this nucleotide base code into the amino acid code. But still, it determines completely how the protein will look like. And in case of glycans, which are also biological polymers, they consist of sugar residues. We don't have an exact template. So each cell kind of, just imagine you have one protein which is glycosylated. The place where the N glycan will be put on this protein is determined by the protein structure, by the sequence of the amino acids in the proteins, but the exact structure of what kind of glycan will be attached to it is not inscribed anywhere. There is no exact template.
Now, to explore this area further, now I think most of us are familiar with the concept of genetic predispositions and genetic diseases. Our proteins, as you said, are produced based on these genes. So it makes sense that if we have faulty genes, we're going to have faulty proteins. Do glycans work in the same way? Can we be predisposed to having bad glycans?
Well, to start, I would say there is no such a thing as a bad glycan per se. Everything is bad or good in a context. So since my research is mainly dealing with glycosylation of IgG, maybe I can bring some example from that field. And of course, so there are the glycans on IgG are very important for the antibody's functions. So the antibody exists not only to recognize the pathogen or some kind of weird kind of foreign proteins in our body. So they also, once they recognize something which is foreign and which has to be eliminated, they also have to notify the rest of the immune system in a way. So there is a special portion of the IgG which is used to transmit the signal to the other components of the immune system. And this is where those glycans occur. And these glycans really determine what kind of the signal will this antibody be sending out to the whole organism. And there are some glycans which kind of skew the response more to the inflammation. So let's start the inflammatory processes, let's get rid of some infected cells, let's kill everything which resembles this pathogenic molecule. And some other responses might be completely different. Okay, let's stop the inflammation. Let's not overactivate our immune system. And depending on the context, this can be bad or good. So if we're talking about some infectious disease, yes, we need to kill all infected cells, we need to get rid of the parasites and so on. But when we're speaking about autoimmunity, when our organism is producing antibodies against our own tissues and our own proteins, of course, excessive inflammation will only kind of lead to the more severe disease. So it's not good in this case. So first, this is what I wanted to say, that we need to think about the context. Whether this glycan is good or not depends on like where we live, what is the current state of affairs, and what we are doing. But yes, indeed, I would say maybe partially due to the fact that some people produce a lot of these pro-inflammatory, to simplify it, glycans, maybe they are more prone to develop stronger autoimmune reactions. And this is partially, of course, due to genetics. We know that glycosylation of IgG, for example, what kind of glycans in which proportions will end up on our IgG molecules, is in a way partially predetermined by our genes. And we know that from the studies on twins, and we know that also from genome-wide association studies. So it has been shown that there are some genes responsible for that.
We are going to get into genome-wide association studies, but thank you for mentioning IgGs. Those are immunoglobulin G, which is a type of antibody. And I want you to explain what makes this antibody so suitable for measuring glycans.
Well, most of all, it is very easy to get enough of this protein, because when we would like to measure and quantify amounts of different glycans attached to some protein, first of all, we need to get the protein itself from some biological sample. IgG is present in big concentrations in the blood serum of healthy people too, because we are always producing some sorts of antibodies throughout our lives. And this is actually one of the most abundant glycosylated proteins that can be found in human bloodstream. So it's very easy to isolate it. That's one reason, but that's not the main reason. The main reason, I guess, is in combination with the first one. As I said, glycans that are found in IgG are extremely important for the IgG's functions. The IgG, which has no glycans, like at all, it's not a functional IgG molecule. It doesn't even fold properly into a conformation that would be able to fulfill its functions. So it makes sense from the biological point of view to learn about IgG's glycans, since they are so important for its functions. And on the other hand, it's also not so difficult. So I guess it was just the first protein that people started to study, like regarding its glycosylation, because it was so available. And it's very easy to sample. It's not, it's invasive, of course, when someone has to give a blood sample, but it's not too bad. And you can get enough even from a small sample of blood. It's usually easier if you're doing it like from the vein, but it's relatively non-invasive. It's not like spinal puncture or something like that.
Now, you have mentioned genome-wide association studies, or GWAS for short. What are they and what are they used for?
So this is one of the recently developed techniques, which became available, of course, when we managed to make sequencing and genotyping of the whole, human genome relatively cheap and relatively easy. So the aim of genome-wide association study is to find out what kind of genes define a certain trait. So it could be like any kind of trait that interests us. It could be height, it could be glycosylation of IgG. Well, there are traits in humans, like in any organism, which are defined only by one gene and like completely defined by this gene. Like I think freckles are supposed to be monogenic. So there is just one gene. You can introduce a mutation into this gene, and then the person who has this mutation won't have freckles. But when we're speaking about height or IgG glycans, there are many genes which are influencing those traits. In case of height, I think it's like thousands of genes. And such traits are called polygenic. And of course, here it's very interesting to try to find all of these genes, at least as many genes as possible that can kind of make an impact on the trait. And GWAS is a very good bioinformatical way of doing that. So you don't have to use these classic methods of genetics when you trace generations from one family, trying to figure out on which chromosome this gene is occurring, judging by different markers and different phenotypes. It takes a lot of time, and in case of humans it's really almost impossible to do. But here what we can do, we can take a sample from a population, like a few hundreds of people, even a few thousands of people, a few hundred thousands of people. We can all check, we don't even have to sequence their genomes. We can just test a few specific mutations, which are distributed, like not a few, like also like millions of them actually, because they have to be spread evenly through the genome. And we can run a statistical test on each of those mutations. Is this mutation, is this particular nucleotide substitution associated with a trait of interest or not? So basically for each of these markers, which is spread over all 46, 23 if you want chromosomes, we will test them whether people who have this mutation tend on average to be, like say, taller or shorter than the average person. So does this particular mutation have anything to do with the trait of interest or not? And of course there is a bunch of statistical techniques associated with the method. Of course I'm like saying, just like roughly how it looks like, because it's not as simple just like let's test all the mutations and be done with that. You need to control for some things when you're selecting your population. You need to be careful with your tests. You need to take into account that some mutations, the closer the genes are in the chromosome, the more they tend to be transferred to the next generation together. So there's like linkage disequilibrium. So it's a bit kind of but in essence it works like this. You test all of these markers over all the chromosomes, see if they're connected to your trait of interest or not. And this was applied to glycosylation of IgG as well. And there were a few genes found, so to say.
That was actually going to be my next question. So how is GWAS applicable for testing the genetic control of glycosylation?
It's pretty applicable, of course, once there were these large-scale glycomic studies. So people, for example, checked the glycans on IgG, their proportions, ratios of different traits in a big number of people, like up to several thousands people, or like, the same for all plasma proteins was done. It was just, logical. Okay, let's try to find the genes which are responsible for that, because this is already enough for a GWAS. So, for the last, I would say, 10 years, definitely we're seeing some studies like that. And as for IgG, I think for now we know already like 30 genetic loci, or like potential genes, so to say, which might be responsible for IgG glycosylation. For plasma now, I think it's a bit less than 30, but it also, like, it depends on how much of say variation that we see in this trait, like in glycosylation of IgG, is due to genetics and how much is due to the environment, and then also how much each particular gene is responsible, so to say, for the trait. And in case of IgG and plasma glycosylation, actually I would say genetics is important, but also apparently the environment is important. Because the genes do not explain, like, all the variation that we see in the glycosylation. So definitely there is some room left for the environment, and every gene explains just a tiny bit, I would say, of all the variability that we see. So to find all of these genes, to locate all of these genes, you need like, the more people you introduce to the study, the more powerful the GWAS is going to be. I guess by this time we're now in this track when we're trying to recruit more and more people and introduce larger and larger cohorts to be able to detect all of those genes. But in fact, it's like up to 30-40 genes, and each of them influences a bit, but still is important.
Now, you said that both genetic and environmental impact are important. Could you explain what you mean by environmental impact?
Every trait, I guess, has a bit of nature and a bit of nurture into that. Every human phenotype, well, almost every. I mean, it depends on which trait we're talking about. But in case of IgG glycosylation, definitely also environment changes. Like for instance, people can get sick. You can get some acute infection, and then of course your IgG profile will change. You can get vaccinated against some disease again. It will influence you and your glycans. And it also is known that almost any physiological process is in some way affecting our IgG glycosylation, because we see that in diseases, we see that in pregnancy, we see that during aging. So obviously it's not only up to your genetic background.
Linking this back to epigenetics, could you quickly explain how environment might play a role in epigenetic changes?
It's actually very interesting. That's maybe the first example that comes to my mind is that learning as a cognitive process is heavily dependent on epigenetic changes. So the moment we are learning some new information, maybe we're learning a new language, maybe we're just learning how to speak, we have to establish some new connections between the neurons in our brain. A lot in our brain physiology has to change. The neurons need to kind of remember to fire up in the response to some certain stimuli that we have never met before. And it is all achieved by changing, methylation of certain regions of the genome. So some genes become active, some become inactive. And since this methylation is not only methylation, all kind of genetic marks which concern the DNA or the proteins around it, they are very mobile. So you can put them and you can reverse them. And this makes it an ideal way for the cell to regulate its own activity and to respond to something new which is coming from the outside, in a way. So learning processes definitely epigenetic, will heavily dependent on epigenetics.
Coming quickly back to GWAS, what did GWAS reveal about heritability of the human glycome?
That's actually kind of interesting, but the GWAS per se cannot tell you much about the heritability. So heritability usually tend to estimate from pedigrees or from the study on twins, dizygotic or monozygotic. And then you kind of can assess, how much of this heritability is explained by the GWAS. Well, in case of glycans, we have some measurements. I really wouldn't like to go into that because this is the part where, I'm not sure that anyone is ever asked, how heritable are the glycans? Well, we asked this question, but when you do these studies on different populations of people, you get different answers. So we know that, yes, indeed, there is some heritable component to that, but how precise are our estimates, I'm not really sure.
Perhaps we have to wait a few years to ask this question again.
Yes, I guess so, because I don't know what we start with. So if you look at studies that were done in these Croatian populations from the islands when they had genealogists and stuff, so they also kind of estimated how heritable are some IgG glycans or some plasma glycans. And then we did the same, for example, for plasma on twins with this Twins UK cohort. And our estimates were not really the same all the time. So yes, we came to the same conclusion that glycans must be heritable, but the extent of this heritability was very different, which might sound surprising, but in the end it is not really, because we know that heritability kind of this concept of how much the phenotype will be able to reveal itself depends on the environment as well. And it also depends on the genetic context in which each mutation is placed. That's for sure. What I mean, it's kind of just imagine, for instance, what you can have a mutation for some disease, for example, which leads to the disease. But if you're in a certain context, this mutation will never lead to the disease itself. I think in Greenland now they have a problem with native people because they have very high incidence of type 2 diabetes, which was never the issue 50 years ago. It turns out they have a mutation which kind of prevents them from effectively utilizing glucose from the bloodstream, effectively transferring it to the muscle cells and to, whenever it needs to go. But the thing is that before they never had a lot of sugars, carbohydrates in their diet anyway, so it wasn't a problem. And now they all switched to the Western culture, to the Western lifestyle, and now they consume a lot of food with a lot of sugars, and suddenly it became a problem. So the same can also happen with the glycans. So maybe if you live in one Croatian island, your environment is such that this particular mutation will never influence your glycans, and they will not appear heritable at all. But when you're talking about a population which lives in a completely different environment, and maybe they also have completely different mutations, it's not the same population, maybe this glycan will appear more heritable in these conditions. but we don't know exactly what the conditions are and how they influence the heritability.
No, of course. I imagine you would need a very, very large cohort to properly study that.
Yeah, and you need to control for a lot of things, and now maybe we're not really even able to say what we have to control, what kind of environmental influences are the most important for IgG glycosylation. Is it smoking? Is it the kind of diet you have?
No, but I think your example about Greenland is really good for our listeners to maybe put everything you've been saying into context, where the same perhaps genetic traits have been passed on for generations, but in the last 50 years the environment has changed to such an extent that only now they're being problematic in terms of their health.
I guess this is like the first thing that they tell you when you're studying evolutionary biology, that all mutations it makes sense only in the context. Sometimes you acquire something useful, but you don't know that it will get useful at some point, and you will be able to use it, only later on when the environment will change and you will have to adapt. And wow, you have already all the means to do that. Or maybe it's the opposite, something that was useful, very much needed in the past. So we evolved to, have this trait. Now it becomes redundant. Like, why we have the problem with obesity in the modern world. Why this is a big issue, like in almost any developed country, because we evolved for thousands of years in a situation when we never had enough food. So the life of our ancestors were, maybe you will be able to kill a mammoth at some point, and you will feed on it, for weeks, and then there will be weeks without any substantial amount of fats and meats and stuff like that. So people survived. Those who survived, they usually were able to accumulate a lot of fat from a small, amount of food. And now the situation is the opposite. We have too much food. And now we're at the disadvantage. Those of us who would be thriving, in prehistoric times are now dealing, you haven't eaten that much, but you're already getting fat. Or, we were also evolving to like sugary food and like salty food, because sugars and salt are things which were actually missing from the diet of, early humans. It would be like beneficial to like those things and to eat them, because you will never have a lot of that. And now we're in our life, everything around us is salty or with a lot of sugar. So again, we are in danger.
I mean, that also explains why it is some of the most dangerous processed foods have excess of sugar and fat, because they are so addictive.
Yeah, we evolved to be addicted to those things. Just, the situation changed and we still haven't had enough time to adapt to that.
Now, bringing it back to the IgG glycome, you already mentioned that it does change in response to environment. Could you maybe give more detail about the circumstances in which it changes and how perhaps it can be changed back, depending on context?
To think of a good example, we know that smoking has an effect on that. In general, like also on plasma glycosylation. And I think there was also a study, it was not the GWAS, but more like EVAS, like epigenome-wide study. So the researchers were trying to see if a certain kind of patterns of methylation are connected with some glycan traits. And they found that yes, indeed, methylation of a few genes is changed, apparently affected by smoking, and then in turn it affects production of the glycans. So people tend to have, like, I think, if I'm not mistaken, more bisected glycans, like this specific structure on your IgG glycan, which is increased in smoking people. But this is actually common knowledge that environment affects our epigenome. So even our diet affects our epigenetic markers, because it affects our gut microbiota, and our gut microbiota metabolizes the nutrients in our guts, and then it kind of also sends some of these byproducts of their metabolism to our cells, and our cells respond to it in a certain way. And what kind of microbes we have in our guts influences the kind of metabolites that we're getting from them. And the other way around, it's also kind of true, vice versa is true. So depending on our epigenetic state, maybe metabolism of our intestinal cells is different, and then they're influencing our microbiota, and they're kind of defining what kind of microbes can colonize our guts and which kinds cannot. So it's not surprising.
Now, as you said, behavior can change our glycans, but if we were to change our behavior, would our glycans revert to a different pattern?
I think this is kind of possible, because I don't think there were a lot of studies on that, but I remember one which was actually coming from Genos. It was coming also from the faculty of the University of Zagreb. So they checked, for example, if exercising will change the baseline levels of glycans in people. So they had, I think, students who were studying physical activities. So like maybe trainers and physical training teachers, future. Really, they all were also sportsmen, so they were familiar with all kinds of exercises. And so they asked one group of people to refrain from some, additional training for a few months, I think. And the other group was having regular intensive training. And they definitely showed that glycosylation of IgG changed in those people who were training. So apparently, if you introduce some differences in your lifestyle, you might end up with different IgG glycans. Also have to remember that it's not, only exercise. There are other factors like infections, infectious diseases, smoking as well, a lot of different habits that you can change.
No, but I think it's very important to start understanding for all of us that our environment, our behavior do shape our health and our glycans as well. Could you maybe explain what pleiotropy means?
So pleiotropy is a situation when you have one gene, but this gene is affecting a lot of different traits in your organism. So maybe sometimes they seem like not connected at all, but in fact they are the results of one gene being mutated. So I think the most striking example of that is the fact that people know that sometimes white cats have problems with hearing. So they can be like partially deaf or completely deaf. And it's very difficult to connect why the coat color has something to do with the hearing abilities. But in fact, this is all the result of the same mutation. So this is the mutation which does not allow some, at the very, very early stages when, the embryo is even developing, migration of some sorts of cells is really faulty. So the thing is, melanocytes and nervous cells, which make these nerves that innervates the inner ear, they actually come from the same embryonic tissue, like part tissue, so to say. And so melanocytes aren't able to migrate to the hair follicles, so the hair has no color. And also the nerves didn't have the time to migrate properly and form the nerves, so the hearing is impaired. And this is actually kind of, most of our traits are controlled, in the end you have some traits which are related, and there is one common regulator which is at the top of this interaction network of all the genes. So if you do something to this main regulator, you will in the end mess up with a lot of different things. There is a good story not about IgG but about plasma, glycosylation in general of plasma proteins. So there is this regulator of transcription, so activity of genes. So it's the story with the hepatocyte nuclear factor 1 alpha, which is actually a regulator of different things in the liver cells. So one of the function, it kind of influences the production of insulin. The other thing, it also regulates fucosylation, antennar fucosylation of any glycan which is produced by this cell. So if there are some mutations, people will have in this HNF1 alpha gene, people won't have enough insulin, so they will start to develop diabetes at some point. It's not exactly type 1 diabetes, it's not exactly type 2 diabetes. It's a very special form of diabetes which is influenced just by mutation in this one gene. On the other hand, the same people will have skewness in glycosylation of their plasma proteins. And this was also discovered in the GWAS. that this HNF1 alpha is responsible for fucosylation, for antennary fucosylation of plasma glycans. And then they realized, oh, this gene is also involved into this specific form of diabetes. So maybe you can use measurements of plasma glycans to detect this sort of diabetes in people. You don't have to do genetic screening, you just do quantification of the glycans in your plasma. And apparently this is working. This is a situation of pleiotropy, so to say. One mutation is influencing two traits.
We are going to come back to glycans as biomarkers for our listeners who might be impatient about that topic. But could you tell me, when it comes to your other glycan studies in this area, which other diseases were found to be pleiotropic?
There are some mutations which is influencing a disease and the glycan trait as well. There was a bunch of them. So if we're speaking about IgG, I think there was a number of autoimmune diseases such as lupus, rheumatoid arthritis, also inflammatory bowel disease, so both Crohn's disease and ulcerative colitis were associated with some kind of pleiotropic with the glycosylation of IgG. And Parkinson's diseases, Alzheimer's, I think low density lipids in the bloodstream as well. There should be some also cardiac phenotypes, although I'm not really, I don't remember by heart whether also, probably, like maybe hypertension or something like that.
When putting all of this into context, would this imply a causative relationship whereby the changes in the IgG glycome lead to disease, or they are caused as a consequence or disease, or do they just seem to be completely unrelated?
That's a very interesting question. And it's surprisingly not so easy to answer. It's, that's the question that we start asking ourselves the moment you say this pleiotropy. So are the glycans causing the disease, or is the disease making IgG glycans to change? Normally, causality, like these causality questions in health and diseases, are answered by setting up a randomized control trial. So you have a group of similar people, and then you randomly assign them to treatment and control groups. So one group will get some special sort of treatment, and then in the end you will compare the health outcome. So did this intervention somehow influenced, if this intervention somehow influenced the disease health outcome in any way. With the glycosylation, we cannot set up a randomized control trial study like that. You cannot really manipulate all the glycans on a person's IgG easily and then see if it leads to differential outcome in, rheumatoid arthritis. Most of all, a lot of people are not even predisposed to have rheumatoid arthritis. So you need to, it's impossible. It's really very difficult. And even if you find a cohort of people who have relatives who have rheumatoid arthritis, how you are going to change their IgG glycosylation? How are you going to manipulate that? There is no easy way to do that in humans, plus, how ethical is that? It's a different question. So what we do, we come to biostatistical, bioinformatical methods, and we try to do that in silico rather than, in a test tube.
Could you explain what silico is in silico?
In silico, it's like when you perform an experiment, not in the wet lab, but you're trying to do that computationally, use some bioinformatical methods. And here again, genome-wide association studies come to help us, because what we can use, the fact, instead of randomly assigning people to control in trial groups, you can use the fact that mutations are distributed also randomly in the population. So you can treat people, okay, if you have this certain mutation, you will be trial group, and if you don't have it, you will be control. And then you can also compare outcome in this to randomly assigned group. But you won't take just any random mutation. You will take mutation which has something to do with the trait that you want to check if it is causing the other trait. So we'll take this mutation which are causing, changes in the IgG glycosylation, and then you will see if these people also have people segregated based on these mutations. If they also have differences regarding, for example, rheumatoid arthritis. If, for example, people who tend to have in this case, if people who are genetically predisposed to have higher bisection on IgG glycans are also more often getting rheumatoid arthritis. So this will be our a bioinformatical proxy for a randomized control trial, and it's called Mendelian randomization. This is also a specific method to study causality between two complex traits. So we did that for IgG glycans and diseases, and here I have to say that it was not so easy to perform such a study, because for Mendelian randomization, your GWAS is from where you derive the information about the effect of genes on different traits, like in this case, effect of genes on glycans. It has to be very powerful. And as I said, genetics, which determines our glycans, is, I wouldn't like to say weak, but you have like really a lot of genes with very small effect on the glycosylation trait. So we were able to detect some effect only in one case. So we found that actually it's not the glycan that was influencing the disease, it was the other way around. So if people who are predisposed to have lupus, to develop lupus, they are also lupus kind of the same genetic predisposition for lupus also skews their glycosylation into having a lot of glycans with bisectin and GLACNAC, so specific glycan structure. But this doesn't mean that there are no other relationships like that. Maybe just our GWAS studies are yet too weak to have enough instruments, like, to have enough of these genetic markers which are connected to glycosylation to test the hypothesis of glycans influence in some diseases properly. So we just maybe need, again, more people in our studies.
While we're still talking about implying causality in diseases of pleiotropic traits, could you comment on why it is important to consider the tissue context?
Well, as I said, because basically different tissues work by different rules. They are governed by different rules. The same gene can be activated by different regulatory networks in different cell types. And the same mutation can also lead to different consequences in different cell types. I think we know that for glycosylation as well, because recently there was a study published on the other plasma protein, on transferrin. So what is the genetic control on transferrin? And you have similar traits, similar kind of glycans that you can find on IgG and on transferrin, but the regulation is completely different. Because IgG is produced like as any antibody in the B cells, while transferrin is a product of liver cells, hepatocytes. So you have mutations in completely different transcription factors, so proteins that kind of activate transcription, regulate the activity of certain genes. But you have two different transcription factors that influence the same glycan trait on two different proteins which are produced by different types of cells. So of course this is like extremely important, the tissue context, at where is your protein actually produced? Because regulation, well, it's obvious that it should be different in different cell types and different organs.
Now stepping away from pleiotropic diseases for a while, I found a study by, I think it was Stambuk et al., very, very interesting, where they looked at how the country you live in impacts the IgG glycome and, by extension, chronic inflammation and biological age. Can you tell me about this study a bit?
Yeah, it was a very interesting study done by my colleagues here in Genos. They checked if there is any connection, as you said, between IgG glycans and different, countries. Are there any interesting dependencies that we're seeing there? I think the most amazing kind of result that they got there, it was that glycans are actually associated with how developed the country is. So there are some statistical indices which show you how developed the country is, and you can kind of correlate glycans with this index like really nicely. Correlation was like really strong. So for instance, it was, I think they found correlations between abundance of this. So on IgG you have mainly glycans with two antennas, and these antennas can be decorated in different ways. So there could be some additional galactose residues put on these antennas. Then on top of that can be sialic acids attached to those galactose residues. And then they see that the more developed the country is, the more people tend to have these galactosylated structures in their glycomes. But if we're talking about still countries which are still developing and where, you know, the standards of living are not as high as in the, so to say, first world countries, you see a lot of agalactosylated structures. So there were some differences.
And what is the importance of galactosylation on these antennas?
Simple answer would be, you know, we always believe that galactosylated structures and cialylated structures are kind of anti-inflammatory. Although now we start to realize maybe it's not that simple. Because like sometimes galactosylated structures can also activate some inflammatory pathways. I think maybe the better example was also the abundance of corfucose on those glycans. Corfucose was also a lot of corfucose was associated with developed countries, while less developed countries tended to have more afucosylated glycans. And again, here we know it's like already almost set in stone that corfucose leads to anti-inflammatory response, while absence of corfucose is strongly pro-inflammatory. And maybe this is connected with the lifestyle. So maybe people from developing countries, so the healthcare is not yet up to the current standards. Maybe they adjust the antigenic load on these people is heavier than people in developed countries. So they live in the places where the healthcare system is not yet working properly. Maybe they just are, living in poverty, so they don't have access to the good healthcare. Maybe they don't have the access to proper nutrition, which is also a factor. Who knows? But in the end, maybe just they need to fight infections, diseases more than people from developed countries. So this is the result. I guess this was the main idea. So maybe this has to do with people from developing countries being more exposed to different pathogens. But you can also think about other explanations because, of course, there are also genetic differences between the populations. So we don't know, was it just the environment that kind of made those people to produce IgG with such characteristics? Or was there also a genetic component to that? We don't really know.
Definitely a very interesting question, though. Now I think we can come back to the topic of IgG glycans as biomarkers. Now, how does the environmental influence, which we've talked about quite a bit in this conversation, now how does this environmental influence of the IgG glycome make them well suited to be diagnostic and prognostic biomarkers?
Since we know that glycans respond to almost everything that happens to us, in our life, we can see the changes in glycans that are connected to any major disease that is happening. We also see the changes in pregnancy, and we even see the changes during aging. So just, we can use these changes. We can use measurements of IgG glycans or plasma glycans to make some judgment about the person's health status. Or there was a great example. I think we should provide the link about that on this test. I think Helena Biosciences are now providing this test for customers, so you can check the certain glycans in your plasma, and based on that they can tell you what is happening to your liver. So whether your liver is healthy, or do you have maybe fibrosis, cirrhosis, or already some hepatocellular carcinoma, which is the terminal stage of the liver damage. And apparently, as you can see, it can not only distinguish between healthy people and sick people, but it can also, your glycans can define at which stage of liver damage you are currently.
We can definitely, if you find the link, we can definitely put it down in the description. When it comes to talking about glycans as biomarkers, we do quite extensively talk about it on this podcast, but it's not very frequently you see it in a clinical setting. Now, what are some of the challenges when developing the screening methods for glycans as biomarkers in a clinical setting?
Usually when you develop any kind of biomarker, it's challenging. So I think First, of course, firstly, when we are speaking about glycosylation, you have to work with some sort of glycans which you can isolate in quantities that are necessary for, reliable measurements. Like that's what you're measuring, you actually are sure that you are really precise. So for instance, with IgG, it is easy because like there is a lot of IgG in our serum. But when you decide, to go to some less abundant proteins, which also can be really crucial to your health, or which can convey a lot of interesting information about what is happening to your body, you need to develop a method how to isolate those proteins, how to enrich them, so that technically you will be able to get, even reliable glycan measurements. But also, as with any biomarker, I guess even more complicated tasks come next, because of course you need, to sell your biomarker, to introduce it into clinics. It has to have some value. So you need to show that indeed this biomarker can replace some commonly used markers, which, for example, are used in diagnostics currently, that your biomarker is better, and like how it is better. Is it maybe like easier to access? Maybe it's a less invasive procedure, or maybe it's way more precise, or maybe it can detect the disease earlier before the onset of symptoms. So it has to have some benefits to switch to our new diagnostic marker. Plus, also, of course, you need to consider can you actually change anything for the patients for being diagnosed with the disease? So when I was talking about this specific kind of diabetes, which was connected with like glycosylation of the glycans, the thing is that those people who have this specific form of this mature onset diabetes of the young, MODY diabetes, which is connected with HNF1 alpha mutations, they need completely different treatment than people who have type 1 or type 2 diabetes. So in this case, it definitely would make sense to recognize those patients as early as possible and give them this specialized treatment, which will help them, because like insulin therapy in the beginning does nothing for them. They are not insulin responsive. Or maybe if you are able to detect some malignant transformation in a person way before the symptoms start to manifest, then you will be in time to save the person's life. So in this case, maybe glycans can help you, because I think it's known for colorectal cancer and also for rheumatoid arthritis that the changes in the IgG glycosylation start way before the person actually starts to feel that something is wrong. So in this case, but you need to research all that. You need to be sure that the marker that you're trying to develop will in the end be useful. Because it's not enough just to say, okay, this thing is different in people with a disease and people who are healthy, and this is enough for a marker. Nope. You need also like commercial estimates, like will it be really cheaper to switch to this new diagnostic system than the previous one? What are the benefits? How the healthcare system will benefit from this new marker? So yeah, a lot of challenges.
No, but definitely thinking about the prognostic and preventative potential of glycan biomarkers sounds very promising for the future. I think a very good place to conclude is by asking you, what would you say are exciting questions in this area which are yet to be answered?
Well, glycobiology still has a lot of unanswered questions, and you can list them for ages. So maybe I can just mention a few topics that are like the most interesting for me, which is very subjective. It's not an objective list. Because my work is connected with regulation of glycosylation of immunoglobulin G and antibodies and glycans in immunity, I'm very interested in how this regulation is happening on the level of the B cell. So what are the exactly biochemical and genetic pathways that connect different transcription factors, different signals that the B cells that produce antibodies receive from outside their maturation, how it is all connected with the kind of glycans that the B cells put on the antibodies that they secrete. So we know that during the process of maturation of a B cell, it undergoes a lot of transformations. So at first it's secreting not IgG, it starts from IgM, which is not even secreted at first, it's just displayed on the surface of the cell as a receptor. Then at some point if a certain conditions are met, a lot of conditions. So this B cell has to recognize some pathogenic protein or glycan. So it has to find the antigen which is suitable for its antibodies. It has to, receive, so to say, support from the other cells of the immunity to be able to proliferate. So how all this unimaginably complex amount of events that have to happen, and how do they all interact with the genetic background of the cell to produce some sort of glycans. So how it all is reflected in the glycans, this combination of environment, genetics, and God knows what else. It's really very interesting and very complex. And but this will be exciting to study, because by now I don't think we even are sure. So if you take one B cell clone which is producing one specific antibody, so are they always putting is some sort of glycan really preferred, or is it more a mixture of different clones that each clone is producing its own glycan, or is it completely random? We're not sure yet. And this would be extremely interesting to answer, and it's very important for the studies of autoimmunity, for instance, because we know that glycans in autoimmune diseases, glycans on the antibodies play an important role. Another interesting question is how glycosylation is regulated at the cellular level. So in any cell, every cell has to glycosylate hundreds of different proteins, and they are all glycosylated in different manners. So what is behind it? Also, an immense number of factors, genome-wide association studies help us to pinpoint the genes and the proteins that might be crucial, but still you have to consider this whole process of glycosylation. Abundance of the building blocks, the sugars, the activity of the enzymes, even the environment in the small compartments of the Golgi apparatus where glycosylation takes place. It's also incredibly complex and incredibly interesting, and it's somewhere at the border between, say, genetics and cytology and biochemistry. Very complex, requires a lot of people from different branches of biology to come together and work together on a complex problem. And maybe the third one that I would like to thing that I would like to mention is host-pathogen interactions. In general, how our cells recognize pathogens. How do we recognize something as a foreign agent? Or how these viruses and bacteria, how they're using our glycans to infect our bodies. Recognition of glycans plays a very important role in host-pathogen interactions. So all the studies that I trying to see on the always check on the web what is new in this in this part of the glycan studies, and it always amazes me, because now this pandemics was happening, COVID was a hot topic, and then we all discovered a lot about glycosylation and antibody responses and how our immunity is using different glycans and how viruses are taking advantage of our glycans. Even COVID is using cellulation on the surface of our cells to bind to the cells and infect. So really, I think we are also waiting for a lot of new discoveries in the glycoscience, for sure.
No, but it sounds like a field to keep an eye on, and to keep updating your knowledge constantly. Thank you very much, Olga, for joining our conversation today.
Thank you very much for inviting me. It was a pleasure.
Now, speaking to our listeners, I hope this conversation gave you an insight into our current understanding of genetic and environmental impact on IgG glycosylation, as well as some of the questions which are yet to be answered. If you would like to access more information about this conversation and Olga's previous work, follow the link in the description to the show notes for this episode. Equally, if you want to find out more about GlycanAge, head on to GlycanAge.com, where you can access a whole list of our scientific publications, blog posts, testimonials, and of course, this is where you can order your GlycanAge test kit. Watch out for our next episode, where I will be joined by Peter Joshi, an actuary turned geneticist who employs his mathematical skills to conduct genomic analyses of complex traits, especially lifespan. We'll be discussing the different hallmarks of aging, as well as how we measure and interpret biological age. Please don't forget to leave ratings and reviews for this episode and engage with us on social media. Thank you for listening and have a great day.